right, we are already on the second topic of Unit 3. This is the first of 3 lectures we're going to do on photosynthesis. This is sort of the intro one. We're going to do a general overview, look a little bit at the evolution, and then finish with a preview of what's to come for the actual biochemical process that will be the focus of our second and third note-taking sessions on photosynthesis. So what is photosynthesis? I know that you already know, right? Like we've been learning about photosynthesis since the 2nd grade. I know that you know that this is the process by which photosynthetic organisms like plants can take sunlight energy and convert it into food. In addition to understanding a lot more details than that at this level, I also want you to think about the process of photosynthesis through the lens of energy. You've recently learned a little bit more about energy, namely that it cannot be created nor destroyed, but it can be transferred and transformed. So we're going to think about how that energy is transferred, for example, from the sun to the plant and along the different steps of this process, and transformed from sunlight energy to chemical energy. And there are little steps along the way and transformations along the way that you need to understand. So that's our new framing of a process that you already know. So who can do this biochemical process? This is also mostly review probably from the ecology that you learned in general biology. Let's actually skip over who can do it and go to the right to see who can't do it. So who can't do it are organisms like us, and like this adorable puppy that I just put on here because I thought we could all stand to look at a puppy. These are the heterotrophs. These are organisms who, like us, must eat other organisms to get our chemical energy. So to get the organic molecules that we need to run our cellular processes, we need to eat food, essentially. If you have to eat food, then you can't do photosynthesis, okay? So that's heterotrophs. The autotrophs, this is the category of organisms that can actually make their own food. So the definition I put up here is that these are organisms that can obtain organic food molecules without eating other organisms, but essentially we're making our own food if you're in this category. Most students assume that all autotrophs are photoautotrophs. So these are the ones at the bottom here, and most of the ones we're going to talk about are definitely in this category. Most autotrophs are photoautotrophs, or autotrophs that convert sunlight energy into chemical energy, like this euglena here, and like plants, most plants. But there is another category that I just want you to be aware of. These are the chemoautotrophs, and these are organisms like this bacteria, P. denitrificans, that can take electrons in their environment and just inorganic molecules, like molecules in their environment, in the dirt, for example, and perform chemical reactions that will turn that into the organic molecules that they need. So that does happen. We're going to be mostly focusing going forward on the photoautotrophs. You should be aware of all of these vocabulary words, and they're all kind of similar. Look at the word parts and like make a couple flashcards because you should know them by heart. In addition to understanding the actual chemical reactions of photosynthesis, you need to understand the context in which those reactions occur. So that means you need to understand where it came from, or the evolution of photosynthesis, and also the relevance of this process to the rest of the biosphere. So like, what ripple effects does this being possible have on the rest of the living world? So we'll get to that later. Let's look at where photosynthesis came from. The first version of this process, a process by which organisms could take sunlight energy and turn it into chemical energy, evolved about 3.4 billion years ago. This graphic suggests 2.6. From what I have understood, I'm not sure if this is maybe a little dated. What I have seen more recently is we're thinking 3.4 billion years ago. Importantly, though, that first version of this process did not release oxygen, which matters because obviously photosynthesis is the main process by which oxygen gets in our atmosphere. And that, like I said, we have to understand the ripple effects. That had a big ripple effect. That process first started around 2.4 billion years ago with cyanobacteria. So I'm showing a picture of cyanobacteria, the different ways that can look. These were the first organisms to do a form of photosynthesis that released oxygen in the air. So you need to be aware of that, but also you should know that these were the guys, that it was cyanobacteria. Just note that these are sometimes called blue-green algae because they can create these sort of algal mats on lakes, for example, but they are really bacteria. They're not algae. Algae are mostly eukaryotes. These are prokaryotic bacteria, the first to do photosynthesis that released oxygen in the air. So how did this process go from a prokaryotic organism into a eukaryotic organism like a plant? In general, when we're talking about photosynthetic organisms, we're talking about plants. And as you well know, those are eukaryotic organisms that are quite different from prokaryotes. So how did that jump occur? You actually already know the answer because we have already learned a little bit about endosymbiosis. Remember, when we were learning about the organelles, we talked about how the common understanding now is that the mitochondrion and the chloroplast evolved from the engulfing of a prokaryotic organism into a eukaryotic organism, and over time, that prokaryote evolving into an organelle for the eukaryote. This is supported by several lines of evidence that you should already be familiar with. One of the things that we're focused on for right now is that the photosynthetic pathways of the prokaryote appear to be sort of the blueprint for the eukaryotic pathways for photosynthesis. So even though there are some differences, there are a lot of similarities, and that's just one of the many similarities between these organelles and their processes and what we assume and think to be is their prokaryotic ancestors. All right, quick detour into plant anatomy. You are not expected to become a botanist, but it is useful to be familiar enough with the parts of the plant so that you can kind of orient yourself when you're understanding the different parts of the process, like what are we doing where, right? Most of the photosynthesis is going to be happening in the leaves, specifically the mesophyll part of the leaf. So there's a little diagram on the right. You can kind of orient yourself there. You're probably already familiar with stomata. In general biology, sometimes we paint the underside of the leaf with nail polish and rip that up to look under the microscope at the stomata. You may remember these are just openings on the other side of the leaf that can open or close to control gas exchange between the plant and the environment. Doing things like, for example, closing in the middle of the day when it's hot, if it's dry, so to protect the plant and like help the plant to keep its water. And then they can open later when we're trying to exchange gases with the environment. There are two vascular tubes that you should be aware of for plants. That would be the xylem and the phloem. The xylem are the water tubes. So those are like the plant vessels that will deliver water to where it needs to go. And the phloem is for glucose or food. In that same diagram, in the red and blue, you can see how these tubes kind of cluster together to form the, quote, veins of the plant, right? The way to remember these, I always tell my students, phloem is for food. That's just like a remembering device to remember that the phloem are the ones that are moving the glucose around, and the xylem is just the other one for the water. And then roots, you know what plant roots do. They absorb water and some other nutrients from the soil. Let's zoom in on one of those plant cells and look at the organelle of the day, the chloroplast. Reminder that you have detailed notes on the anatomy of the chloroplast in Unit 2, Notes 2. That's the second cell tour, so I encourage you to look over those again. But as a refresher, we have a double membrane on the outside. Inside of the inner membrane, we have a watery stroma, and then floating in the stroma are those stacks of membranous discs called thylakoids. Remember, each individual one, looks like a penny, is a thylakoid, and the stack is called a granum, plural grana. When you're thinking about the anatomy of the chloroplast, I would like for you to keep in mind the general themes of surface area, increasing surface area, allowing you to increase the amount of chemical reactions that are occurring, and compartmentalization or separating spaces. So not just right this moment, but as we go through the whole process, think about how those two things are allowing the plant to maximize sugar production. Okay, we have two sets of reactions. The first set is called the light-dependent reactions, sometimes called the light reactions. These occur on and across the thylakoid membrane. The second set of reactions happens in the stroma. This set of reactions, unfortunately, has three names that I'd like for you to be familiar with. Sometimes called the light-independent reactions, once in a while called the dark reactions, and often called the Calvin cycle. This is that second set of reactions where we're actually getting the sugar. So the first set is sort of teeing things up. Second set, we're making the sugar. This happens in the stroma. All right, so that's where eukaryotic cells are doing photosynthesis, but I told you that bacteria, like cyanobacteria, can also perform photosynthesis, and as you well know, they don't have membrane-bound organelles like chloroplasts. So what is the deal? They have evolved a special inner membrane that is also called a thylakoid. Notice that this inner membrane is also exploiting surface area to maximize efficiency. That's where their light reactions are going to occur. And they do their light independent reactions, or their Calvin cycle, in the cytosol. So that's the bacteria version of this. And finally, before I leave you, a preview of where we're going next, which is the actual biochemistry. This is the overall formula or simplified formula for photosynthesis. And you should have a deep understanding of this before you try to get into the details of all the intermediary steps. And what I mean by that is just understand what's going in and what's coming out. So let's look at that. You know that plants, quote, breathe in carbon dioxide, right? So that is a reactant. Plants need water. You know that if you don't water your plant, or plant doesn't have access to water, it'll die. And this is why, because it needs it to perform photosynthesis. So water is another reactant. Light energy is required. Sometimes you'll see it shown like this on the left, or sometimes you'll see it shown as like going into the arrow because it's not technically a reactant, but light energy is obviously required. And then what's coming out as a product? You may not be familiar with that chemical formula, but that's just glucose. Now that I say that, you probably recognize it as a carbohydrate type formula, right? So that's glucose. That's the food that the plant is making, or the photosynthetic organism is making. And you may remember that plants, quote, breathe out oxygen. So that is the other product. All right, great job, you guys. We will see you in class.